This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wondered if evolution can run in reverse? Join us as we unravel the mysteries of Dollo's law, a principle that explores whether lost traits can reappear. In simple terms, we'll delve into the fascinating world of trait reversal, examining intriguing examples from the animal kingdom and the molecular mechanisms that govern these evolutionary U-turns. Get ready to challenge your assumptions about the directionality of evolution and discover the remarkable plasticity of life. Chapter 1. The Curious Case of Lost and Found. Exploring the mystery of trait reversal. Picture this: a prehistoric world teeming with life, where evolution's grand experiment unfolds. In 1893, amidst this backdrop of discovery, a Belgian paleontologist named Dollo proposed a compelling idea about the nature of evolutionary change. He stated that an organism never returns exactly to a former state, even if it finds itself placed in conditions of existence identical to those in which it has previously lived. In essence, Dollo was suggesting that evolution, like time itself, marches forward, leaving its footprints indelibly etched in the history of life. This became known as Dollo's law, a principle that has sparked debate and fueled scientific inquiry for over a century. Imagine a trait, a characteristic that helps an organism survive and thrive. Perhaps it's a physical feature, a behavior, or even a molecular mechanism. Now, imagine that trait disappearing over generations, lost to the winds of evolutionary change. Dollo's law suggests that this lost trait, in its original, precise form, is highly unlikely to reappear, even if environmental conditions shift back to favor it. Evolution prefers to tinker with existing structures, modifying and repurposing them rather than rebuilding lost features from the ground up. Let's take a more concrete example. Think of birds, those feathered descendants of ancient reptiles. Their ancestors had teeth, like most reptiles do. But over millions of years, birds lost their teeth, evolving beaks of various shapes and sizes perfectly suited to their diverse diets. Some beaks are sharp and hooked for tearing flesh. Others are long and slender for probing flowers, and still others are short and stout for cracking seeds. While birds have developed an astonishing array of beak adaptations, none have re-evolved teeth made of the same material, hydroxyapatite, as their reptilian forebears. A 2009 study delved into the molecular mechanisms that might underpin Dollo's law. Researchers examined the evolution of a hormone receptor, a protein molecule that binds to specific hormones, triggering a cellular response. They found that the receptor's shift from binding two hormones to specializing in just one involved changes in its amino acid sequence, the building blocks of proteins. These changes not only altered the receptor's function but also affected its stability. Trying to revert the receptor back to its ancestral state proved difficult because the accumulated changes had made the original configuration less stable. It's like trying to put a puzzle back together after some pieces have been altered; they might not fit anymore. So, does Dollo's law hold true in every case? As we'll explore in the next chapter, there are intriguing cases where evolution seems to defy this principle. Chapter 2. Whispers of Resurrection. When evolution seems to run in reverse. Scattered throughout the natural world are whispers of resurrection, instances where evolution seems to play in reverse. These intriguing exceptions ignite a debate that compels us to re-evaluate our understanding of evolutionary trajectories and the constraints that shape them. One such whisper echoes from the amphibian world. Most frogs, as we understand their evolutionary history, lost teeth on their lower jaw millions of years ago, retaining them only on the upper jaw. This dental arrangement serves them well, allowing them to grasp prey before swallowing it whole. However, Gunther's marsupial frog, a denizen of South American forests, breaks this pattern, sporting teeth on both its upper and lower jaws. Phylogenetic analyses, using genetic data from over 170 amphibian species, suggest that Gunther's marsupial frog re-evolved lower teeth after an estimated 220 million years of their absence in the frog lineage. This re-evolution, however, is not a simple rewind. The regained teeth, while functionally similar, may have a different developmental and genetic basis than those of the frog's ancient ancestors. The insect world also offers its share of evolutionary enigmas. Stick insects, masters of camouflage, have repeatedly lost and regained wings throughout their evolutionary history. This back-and-forth transition, observed in multiple stick insect lineages, suggests that the genetic and developmental architecture for wings can be retained, even after millions of years of flightlessness. The re-evolution of wings, however, is not always a perfect replica of the ancestral form. Perhaps one of the most remarkable examples of trait reversal comes from the fossil record of kangaroos. A study on extinct kangaroo lineages uncovered the reappearance of a molar tooth crest, a feature believed to have been lost millions of years earlier. This reanimation of a seemingly lost trait suggests that evolution can sometimes tinker with the way features are assembled during embryonic development, reactivating dormant genetic pathways. These intriguing cases of trait reversal raise fundamental questions about the interpretation and validity of Dollo's law. Some researchers argue that the law, in its strictest sense, refers to the statistical improbability of exactly retracing an evolutionary pathway. Others, like the late Stephen Jay Gould, suggested that Dollo's law reflects the constraints imposed by evolutionary history. Researchers have proposed various loopholes to explain apparent violations of Dollo's law. One such loophole involves the concept of dormant genes. Genes that are no longer expressed can remain in an organism's genome for millions of years, accumulating mutations but retaining the potential for reactivation. Another loophole involves homologous genes, genes that share a common ancestry but have diverged in function. A gene that is currently used for one purpose can, through mutation and selection, be repurposed to perform a different function, potentially mimicking a lost ancestral trait. The debate surrounding Dollo's law also highlights the challenges of reconstructing evolutionary history. Phylogenetic analyses, the methods used to infer evolutionary relationships, are based on interpreting patterns of shared characteristics among species. However, these patterns can be misleading, especially when dealing with traits that have been lost and regained multiple times. Some researchers argue that certain reported violations of Dollo's law are artifacts of inaccurate phylogenetic reconstructions rather than genuine reversals. A study on protein structure evolution provided a new perspective on Dollo's law. Researchers found that seemingly neutral mutations, changes in a protein's amino acid sequence that don't affect its function, can accumulate over time and destabilize the ancestral protein structure. This destabilization can make it difficult to revert to the ancestral state, even if the original mutations are reversed. The whispers of resurrection challenge our understanding of evolutionary processes and the constraints that shape them. They force us to re-evaluate Dollo's law, not as an absolute dictum, but as a principle that must be interpreted in light of the complex interplay of genetics, development, and environmental pressures. Chapter 3. The Ghosts of Evolution Past. How genes remember what we forgot. Gene silencing, the process by which a gene's activity is suppressed, can occur through various mechanisms. One common method involves changes in gene regulation, the intricate system that controls when and where genes are turned on or off. Imagine a dimmer switch controlling a light. The light bulb, the gene, is still there, but the dimmer switch, gene regulation, can reduce or eliminate its output. Similarly, modifications to the DNA sequence itself, such as mutations in the gene's regulatory regions, can also silence a gene. Reactivation of these dormant genes can be triggered by changes in environmental conditions or mutations that alter gene regulation. For instance, some mammals, including guinea pigs, have lost the ability to synthesize vitamin C due to the inactivation of the gene for L-galonolactone oxidase, the enzyme responsible for the final step in vitamin C production. However, it's conceivable that under certain selective pressures, perhaps a diet consistently lacking in vitamin C, mutations could reactivate this dormant gene, restoring the ability to produce this essential nutrient. Another striking example involves the re-emergence of hind limbs in snakes. Snakes, of course, evolved from four-legged reptiles, and some retain vestigial hind limb structures. While the full development of functional hind limbs in modern snakes is unlikely, it's not entirely beyond the realm of possibility. Mutations affecting developmental pathways, combined with environmental pressures favoring locomotion with limbs, could theoretically reactivate dormant genes and developmental pathways involved in limb formation. The fate of dormant genes over time depends on a delicate balance between preservation and degradation. If a dormant gene remains completely unused for millions of years, it will likely accumulate mutations that render it non-functional, effectively erasing the blueprint from the archive. However, if the dormant gene retains some low level of activity, or if it's involved in other, less obvious functions, it may be preserved for longer periods, increasing the chances of its eventual reactivation. The degree of preservation also depends on the organism's generation time and population size, factors that influence the rate of genetic drift and mutation accumulation. Distinct from dormant genes are atavisms, the spontaneous reappearance of ancestral traits in individual organisms. These are not due to the reactivation of dormant genes in the entire species but rather result from disruptions in normal development during an individual's lifetime. A classic example is the occasional birth of whales with small, hind limb-like structures, a throwback to their four-legged mammalian ancestry. These atavisms are like glitches in the developmental program, revealing the hidden potential for ancestral traits to re-emerge. Atavisms can manifest in a variety of organisms and traits. Horses, for example, can sometimes be born with extra toes, a reminder of their multi-toed ancestors. Insects may exhibit ancestral wing patterns or coloration, and humans can occasionally be born with a tail-like appendage. The frequency of atavisms varies depending on the trait and the organism, but they are generally rare occurrences. The genetic and developmental basis of atavisms lies in the intricate network of genes that orchestrate an organism's development. During embryonic development, a precise sequence of genetic instructions guides the formation of tissues, organs, and body structures. Disruptions in this process, such as mutations in regulatory genes or environmental influences, can cause the expression of ancestral traits that are normally suppressed. It's like a skip in a record, causing the developmental program to momentarily revert to an older version. Developmental pathways, the intricate series of genetic and cellular events that shape an organism's form and function, play a critical role in the re-emergence of ancestral traits. Even small changes in these pathways, especially during embryonic development, can have profound effects on adult morphology. The re-evolution of shell coiling in slipper snails, for instance, involves changes in developmental timing, a phenomenon known as heterochrony. By altering the rate or timing of shell growth during development, these snails have regained a coiled shell from a limpet-like ancestor. Environmental factors can also influence developmental pathways. Temperature, nutrient availability, and exposure to certain chemicals can all affect gene expression in developmental processes, potentially leading to the re-emergence of ancestral traits. This interplay between genes and environment underscores the dynamic nature of development and its role in evolutionary change. The concept of modularity in development further complicates the picture. Development is often organized into modules, semi-independent units that control the development of specific structures or body parts. Changes in gene regulation within these modules can facilitate evolutionary reversals by allowing for the redeployment of existing developmental programs in new contexts. It's crucial to remember that even when traits seem to reappear, evolution rarely, if ever, returns to the exact same prior state. As Dollo's original formulation of the law emphasizes, the organism always keeps some trace of the intermediate stages through which it has passed. The re-evolved trait may be similar in function and appearance to the ancestral trait, but it will have a different evolutionary history, reflecting the unique path that the lineage has taken. The ghosts of evolution past may return, but they do so with a new twist, a testament to the ever-changing nature of life on Earth. Chapter 4. The Unraveling of Dollo's Law, a scientific saga of certainty and doubt. Our story begins in 1893. A time when paleontology was in its nascent stages, unearthing the secrets of a prehistoric world. Louis Dollo, a Belgian paleontologist meticulously studying iguanodon fossils, proposed a seemingly simple yet profound idea. An organism never returns exactly to a former state. This statement, born from observations of fossilized bones and the perceived directionality of evolutionary change, became known as Dollo's law. Initially, it was embraced as a fundamental truth, an inviolable principle akin to the laws of physics. The mid-20th century witnessed a paradigm shift in evolutionary biology with the rise of genetics and the modern synthesis. The discovery of DNA's structure and the mechanisms of inheritance provided a new lens through which to view evolutionary change. Dollo's law, once considered an absolute truth, was reinterpreted as a statement about probability, not impossibility. The focus shifted from the absolute irreversibility of traits to the statistical improbability of retracing the exact same evolutionary trajectory. This reinterpretation was heavily influenced by the concept of mathematical probability. Imagine the vastness of evolutionary space, a multidimensional landscape of possible genetic combinations and developmental pathways. The chances of a lineage precisely replicating a series of random mutations and environmental pressures to regain a lost trait become vanishingly small. This probabilistic view of Dollo's law acknowledged that while exact reversals are highly unlikely, they are not strictly impossible. Further complicating the picture is the phenomenon of acquisition bias. When studying evolutionary reversals, researchers often focus on groups of extant species that exhibit variation in a particular trait. This focus can inadvertently skew our understanding of reversibility. If we study a group of snails where some have coiled shells and others have limpet-like shells, we might be tempted to conclude that the limpet-like form represents a loss of coiling and that some lineages have re-evolved coiling. However, this conclusion depends on correctly identifying the ancestral state. If the limpet-like shell was actually the ancestral condition, then the coiled shell would represent the derived trait, and there would be no question of reversal. Acquisition bias can lead to misinterpretations of ancestral states and, consequently, to erroneous claims of evolutionary reversals. The re-evaluation of Dollo's law has also involved recognizing different levels of reversibility. It's crucial to distinguish between the re-evolution of a similar trait and the precise replication of the original form. The re-evolution of shell coiling in some slipper snails doesn't necessarily mean they regained the exact same coiling mechanism as their ancestors. The coiled shell may have reappeared through a different developmental process, utilizing different genes and molecular pathways. Similarly, the re-emergence of lower teeth in Gunther's marsupial frog, after over 200 million years of absence in the frog lineage, might involve different genetic and developmental mechanisms than their ancestral counterparts. While the trait, lower teeth, has reappeared, the underlying form, at the molecular and developmental level, may be quite different. Stephen Jay Gould, a prominent evolutionary biologist and paleontologist, offered a nuanced interpretation of Dollo's law. He argued that while exact reversals are improbable, evolutionary history does constrain future possibilities. Once broad morphological forms emerge, certain evolutionary pathways become effectively closed off. The adoption of a reptilian body plan, with its characteristic skeletal structure and physiological adaptations, forecloses certain evolutionary options that might have been available to the lineage's ancestors. Gould's interpretation emphasized the importance of historical contingency in shaping evolutionary trajectories. Dollo's law also found a practical application in phylogenetics through the concept of Dollo parsimony. This method assumes that a characteristic is gained only once in a lineage and, if lost, is never regained. This principle is applied to both morphological traits and molecular events, such as gene losses. For instance, the evolution and loss of teeth in vertebrates can be modeled using Dollo parsimony. Teeth, made of hydroxyapatite, are assumed to have evolved once at the base of the vertebrate tree. A 2009 study on protein structure evolution provided a molecular mechanism that could explain the difficulty of evolutionary reversals. Researchers examined a hormone receptor protein that had evolved from an ancestral form capable of binding two hormones to a derived form specific for a single hormone. They found that subsequent neutral mutations, changes that didn't affect hormone binding, had destabilized the ancestral protein structure. Attempting to revert the protein back to its ancestral state, by reversing the original functional mutations, proved challenging because the accumulated neutral changes had made the ancestral configuration unstable. This study highlighted how seemingly inconsequential changes can have profound implications for evolutionary reversibility. In conclusion, while Dollo's law isn't a strict, inviolable law like those of physics, the underlying principle of improbability remains important. The re-evolution of traits, while documented in several cases, rarely involves the precise replication of ancestral forms. The debate surrounding Dollo's law underscores the dynamic nature of scientific inquiry, where established theories are constantly challenged and refined in light of new evidence. Our understanding of evolutionary reversals has evolved from a view of absolute irreversibility to a more nuanced perspective that considers probability, historical contingency, and the intricate interplay of genes, development, and environment. This sets the stage for our next chapter, which will explore the specific genetic mechanisms that underlie these fascinating instances of apparent trait reversal. Chapter 5. Beyond Dollo, the future of evolutionary reversibility. Our understanding of evolutionary reversibility is in constant flux, propelled by ongoing research in phylogenetics, genomics, and evolutionary developmental biology. We're moving beyond simply cataloging apparent reversals and delving into the intricate mechanisms that make them possible. This shift in focus is revealing a more nuanced and complex picture of evolution, one where the past isn't always a reliable predictor of the future, and where the seemingly impossible can sometimes occur. One avenue of exploration focuses on the evolution of proteins, the molecular workhorses of life. The epistatic ratchet effect, where neutral mutations constrain future evolutionary pathways, offers a compelling explanation for why some reversals are so improbable. It's like trying to walk back up a descending escalator. Each neutral mutation makes the return journey more challenging. The example of parasitic wasps provides another glimpse into the complexities of reversibility. These wasps appear to have lost and regained the ability to synthesize fat multiple times throughout their evolutionary history. Research suggests this isn't a case of complete gene loss and regain but rather a switching mechanism. The ability to synthesize fat hasn't vanished entirely, it's simply turned on or off depending on the fat content of the wasp's host insect. This plasticity, the ability to adjust traits in response to environmental cues, highlights the importance of context in understanding trait evolution. Beyond parasitic wasps, the natural world offers a plethora of intriguing examples. Consider the common lizard, a species that exhibits both oviparous, egg-laying, and viviparous, live-bearing populations. Genomic analyses have revealed a surprising twist. Some oviparous lineages appear to have re-evolved egg-laying after their ancestors had transitioned to live birth. This reversal, a significant evolutionary leap, challenges the traditional view that such complex reproductive shifts are irreversible. The re-evolution of oviparity in these lizards underscores the potential for reanimating genetically mothballed features, where dormant genes and developmental pathways can be reactivated under specific ecological pressures. The story of shell coiling in gastropods, particularly slipper snails, offers another perspective on reversibility. Many gastropod lineages have simplified their shells over time, losing the ancestral coiled form in favor of a limpet-like shape. However, some slipper snail genera have seemingly defied this trend, regaining a coiled shell from a limpet-like ancestor. This re-evolution, however, isn't a simple rewind. It likely involves heterochrony, changes in the timing of developmental events, rather than a precise replication of the ancestral coiling mechanism. The debate surrounding this example highlights the difficulty of interpreting evolutionary history and the potential for mistaking convergent evolution, where similar traits arise independently in different lineages, for true reversal. The robustness of phylogenetic trees, the diagrams used to represent evolutionary relationships, also plays a crucial role in these interpretations. A slightly different branching pattern can lead to drastically different conclusions about trait gain and loss. Even at the molecular level, seemingly simple traits can exhibit surprising complexity. The loss and potential regain of teeth in vertebrates provides a classic example. While teeth have been lost multiple times throughout vertebrate evolution, in birds, turtles, and seahorses, for instance, the underlying genetic and developmental pathways may be retained to varying degrees. The re-evolution of teeth, if it were to occur, might not involve the same genes and developmental processes as the original teeth. The primate lineage, including humans, offers another fascinating case study, this time involving muscle evolution. Research has revealed multiple instances of muscle reversions, where muscles lost in ancestral lineages have reappeared in later descendants. A specific example involves the rhomboideus muscle, which connects the shoulder blade to the spine. In some primate lineages, this muscle split into two distinct muscles, only to fuse back into a single muscle in the human lineage. This seemingly minor anatomical detail provides a glimpse into the complex evolutionary history of our own musculature. These diverse examples highlight the challenges in studying and interpreting evolutionary reversals. Distinguishing true reversal from convergent evolution requires careful analysis of phylogenetic relationships, combined with detailed studies of development and genetics. The robustness of phylogenetic trees, the diagrams used to represent evolutionary relationships, is crucial for accurate interpretations. A slightly different branching pattern can lead to drastically different conclusions about the evolutionary history of a trait. The implications of evolutionary reversibility extend beyond academic curiosity. They touch upon fundamental questions about the predictability and directionality of evolution. Is evolution a deterministic process, where the same outcomes are inevitable given the same starting conditions? Or is it contingent, where chance events and historical accidents play a significant role? The study of reversals suggests that evolution is a complex interplay of both contingency and determinism. While the vastness of evolutionary space makes exact reversals improbable, the re-evolution of similar traits, through different pathways, demonstrates the remarkable adaptability and resilience of life. This evolving understanding of reversibility also has practical applications. In medicine, for example, knowing that bacteria can re-evolve antibiotic resistance after losing it has profound implications for developing effective treatment strategies. Rather than simply trying to eliminate resistance, we might need to focus on managing its evolution, slowing down the re-emergence of resistant strains. In conservation biology, recognizing the potential for lost traits to reappear could inform efforts to restore endangered species and manage ecosystems. If a species has lost a crucial adaptation, understanding the mechanisms of trait re-evolution could offer hope for its eventual recovery. The ongoing exploration of evolutionary reversibility is reshaping our understanding of life's history and the evolutionary process. It's a story of surprising twists and turns, where the past isn't always a reliable predictor of the future, and where the seemingly impossible can sometimes occur. By unraveling the genetic and developmental mechanisms that underlie these evolutionary resurrections, we gain a deeper appreciation for the remarkable flexibility and resilience of life on Earth. Dollo's law, the idea that evolution is irreversible, faces challenges from instances of trait re-evolution. While precisely retracing evolutionary steps is statistically improbable, organisms can regain lost traits through different genetic and developmental pathways. This highlights evolution's remarkable plasticity and has implications for fields like medicine and conservation. Where do you think research on evolutionary reversals will lead us? Share your thoughts and questions below. Like, subscribe, and suggest future topics. Never stop exploring the wonders of the natural world.